Here's an overview of gluconeogenesis. Gluconeogenesis is one of the major metabolic pathways in our body, and gluconeogenesis is primarily going to occur at times when we're not consuming any meals. For example, when we are sleeping, there are two main pathways that help us that help our body with providing energy, and one of them is glycogenolysis and the other is gluconeogenesis. So 50% of the energy that comes during our sleep is from glycogenolysis, and usually the other 50% is from gluconeogenesis. When our glycogen stores are depleted, gluconeogenesis is going to be the major metabolic pathway that is going to provide our body with the energy that it needs. And this is especially important because the final product of gluconeogenesis is glucose, and this is going to make sure that our body is not in a hypoglycemic state, because hypoglycemia can lead to coma and even death. So it is why it is crucial that gluconeogenesis is working properly in our body. So at the end of glycolysis, pyruvate is produced, and as you can see here, pyruvate can also be generated through lactate as well as glucogenic amino acids. So one of the other functions of gluconeogenesis is to make sure that lactate does not build up. Lactate is produced by our muscle cells as well as red blood cells, and lactate can then be used in gluconeogenesis to ultimately be converted into glucose. As you can see here, glycerol is also used in the gluconeogenic pathway, and glycerol can enter the pathway through DHAP, and then it can enter at the level of glycerol diethylphosphate as well as fructose 1,6-bisphosphate. Now, as we said, gluconeogenesis is going to mainly occur during times of fasting. So during times when we're not consuming any meals more than after 4 hours. And when we're not consuming anything, then our blood sugar levels are going to be low. And if our blood sugar levels are low, then our insulin levels are also going to be low. Why? Because if our blood sugar levels are low, then our body does not need to produce insulin to take in any extra glucose found in our blood to our cells. And if our insulin levels are low, if you take a look at fatty acid breakdown, if our insulin levels are down, then fatty acid breakdown is going to be upregulated. Why? Because the major hormone or the major enzyme that breaks down triglycerides is hormone-sensitive lipase over here. And as you can see, hormone-sensitive lipase is inhibited by insulin. And during times when we're not eating, our insulin levels are going to be low, and this pathway is going to be upregulated through hormone-sensitive lipase. So hormone-sensitive lipase is going to break down triglyceride into fatty acids and glycerol. And as we said, glycerol can then be used in the gluconeogenic pathway, and it can enter gluconeogenesis at the level of glycerol diethylphosphate as well as fructose 1,6-bisphosphate to ultimately provide us with glucose. Now, fatty acids are going to ultimately result in high acetyl-CoA levels, and as we can see from our image here, fatty acids are going to produce high levels of acetyl-CoA, and acetyl-CoA is actually going to upregulate gluconeogenesis. So if you have high acetyl-CoA levels, it is going to upregulate the enzyme pyruvate carboxylase, converting pyruvate into oxaloacetate, and you can consider this the first step of gluconeogenesis. So to summarize, we said that when we're not eating, our insulin levels are going to be low. If our insulin levels are low, then fatty acid breakdown is going to be upregulated, and we're going to have high levels of fatty acid as well as glycerol. We said that glycerol can be used in the gluconeogenic pathway. It can enter gluconeogenesis at the level of glyceraldehyde 3-phosphate as well as fructose 1,6-bisphosphate. And we said that fatty acids are ultimately going to result in high levels of acetyl-CoA. And we said that acetyl-CoA is going to upregulate the enzyme pyruvate carboxylase, converting pyruvate into oxaloacetate. Now, oxaloacetate needs to be transported into the cytosol for this pathway to continue, and we are going to do this by converting oxaloacetate into malate. And malate can be transported to the cytosol through the malate shuttle, and once it is transported to the cytosol, it can then be converted back into oxaloacetate. And then oxaloacetate can be converted into phosphoenol pyruvate. And the enzyme that does this is phosphoenol pyruvate carboxykinase, or also known as PEPCK. Now, this step is going to require energy in the form of GDP. And if you remember from the citric acid cycle, the conversion of succinyl-CoA to succinate is going to produce GDP, and we can use that GDP over here to help with this step. So now we have phosphoenol pyruvate. Phosphoenol pyruvate can ultimately be converted to glyceraldehyde 3-phosphate as well as fructose 1,6-bisphosphate. Now, in order for fructose 1,6-phosphate to be converted into fructose 6-phosphate, we need the enzyme fructose 1,6-bisphosphatase, as you can see here. And this enzyme is going to be inhibited by fructose 2,6-bisphosphate. And in order for this gluconeogenic pathway to continue, we need low levels of fructose 2,6-bisphosphate so that it does not inhibit the enzyme fructose 1,6-bisphosphatase, so that fructose 1,6-bisphosphate can be converted into fructose 6-phosphate. Now, the way we're going to get around this is with the help of the hormone glucagon. Glucagon is the major catabolic hormone in our body, and glucagon is released in times of fasting. So when we're not eating anything, glucagon is going to be released, and the reason why it is released is to help our body get the energy that it needs during times of fasting. And so when glucagon is released, it is going to upregulate the enzyme fructose 2,6-bisphosphatase. And when it upregulates the enzyme fructose 2,6-bisphosphatase, then fructose 2,6-bisphosphate can be converted into fructose 6-phosphate, resulting in lower levels of fructose 2,6-bisphosphate. If we have low levels of fructose 2,6-bisphosphate, then we're going to have higher levels of fructose 1,6-bisphosphate. And with having high levels of this enzyme, we can then continue the pathway by converting fructose 1,6-bisphosphate into fructose 6-phosphate. After this conversion, we can then convert fructose 6-phosphate into glucose 6-phosphate over here, and then glucose 6-phosphate can be converted into glucose with the help of the enzyme glucose 6-phosphatase. And as you can see here, glucagon is also going to help us with the conversion of glucose 6-phosphate to glucose. And the way it does this is by upregulating the enzyme glucose 6-phosphatase. And so ultimately we get glucose to provide our body with the energy it needs at times when we are not consuming any meals.